Hello everyone, and thank you so much for joining me for today's class. Today we are going to look into container closure system, their characteristics and their testing as per the UPS. So let's go over an overview of the container closures. Okay, we use these containers for the products that we make in the pharmaceutical industry. So this is normal that these products must be, these containers must be safe in a way that it's also going to restrict access to patients who are younger, and they may also be exposed to some risk for an overdose. But also, if there is an interaction in between the material of the container and the drug itself, or the drug coating, for example, this is not good. This is not safe. So we need to go over several requirements in order to make containers and, of course, container closures, so that we're going to make sure that the products that we're giving to the population is safe. So a container closure system is the sum of packaging components. So it's not just one layer. You may have several layers of containers that together contain and protect the dosage form. That's why you will see different materials that we use. You are going also to see that we are going to have different shapes, different structures, and everything has been tested. It means that we don't want to have an interaction between the drug and the containers neither. I'm talking about the chemical, for example, interactions. And we also want that the container closure is the best fit for the use of the drug. For example, we may have some container closures that we are going to refrigerate. Some others we don't. Some of them, they must have some type of limitations for each dose. And here we are going to look into inhalers, for example. So I will continue, of course, on these topics in the next slide. So it may include primary and secondary packaging components. We also have blisters, for example. So they can vary widely in their appearance and physical form. Okay, so what we are going to see today, we are going to look into a few regulations, guidance documents, and of course USPs. And we are going to look into the FDA warning letters. We will go over the types of materials, the attributes for the container closures, such as suitability, the protection, compatibility, and then we'll go over testing methods, where we usually talk about two types of methods, deterministic and probabilistics. Okay, so first of all, a packaging system. What is that? This is a container closure system. So we need to refer to guidance for industry from FDA as of May 1999, and the title is Container Closure Systems for Packagings, Human Drugs and Biologics. So it refers to the sum of packaging components that together contain and protect the dosage form. And again, as I already mentioned, this may include primary and secondary packaging components. Now, we need to know the following terms for the next slides. The first term is container closure system that I already mentioned. This is a sum of packaging components that contain the dosage form. The packaging component is any single part of a container closure system. So typical components are containers like ampoules, vials, bottles, or we can have container liners like tube liners. We can have closures like screw caps or stoppers. We will have closure liners, inner seals, administration ports, and labels. Of course, in different materials, different shapes, different colors, obviously. And they must all have been tested before we use them as container closure systems. Another term is primary packaging component and secondary packaging component. So the primary packaging component is packaging component that is or may be in direct contact with the dosage form, as opposed to the secondary packaging, where it may not be in direct contact with the product. We also have the term suitability. which is these are tests and studies used and accepted for the initial qualification of a component or container closure system for its intended use. And the last term is the quality control. So the tests used and accepted to establish that after the application is approved, the container closure system has the characteristics established in suitability studies. So here we talk about application. The application is, for example, new drug application for the US FDA. It may be new drug submission for Health Canada in Canada. It may be the marketing authorization application for the European Union, so for the EMA. So here, the information which is here, it must be in a specific module, which is module 3 of the CTD, or Common Technical Document backbone. So let's go over the ICH guidelines now. This is ICH Q8, revision 2, discussing container closure systems. So when we go to the ich.org website and we go to the section Q for quality, we will be able to open this, again, guideline, where we talk about the drug substances, about the excipients. There we talk about the drug product. So there's a formulation development, of course, overages, then physicochemical and biological properties. Then it has a chapter related to the manufacturing process development, the container closure system, of course, which is very important to explain, to test, and then to document, and then to add it, obviously, to the CTD backbone of our registration application. And obviously, we also have information about the microbiological attributes and compatibility. So this is, in a nutshell, the different sections that we can see in ICH Q8. Now let's go over the container closure system example. So I'm starting from left and I will continue to the right of these closure systems. The first one is the IV or intravenous bag. This is the format that you see. This is in plastic. Of course, this is in not just a plastic, but it can be, again, it must be a plastic which is already tested. Then we have ampoules. Ampoules are, again, they have a flame sealed tip. And of course, we are going to talk about the ampoules as well. We can also have serum vials. So those serum vials, they have stoppers that are sealed to vial by a crimpled cap that we cannot see here. So we remove the crimpled cap, and then we are going to, of course, use this, I would say, this stopper. Sometimes we remove it. Sometimes we just use a syringe, and we are going to, I would say, penetrate the needle of the syringe through the stopper, which is sealed. Then we also have cartridges. So these are, again, crimp cap that is not shown on the top, and you also have a seal, as you see when you compare with the serum vial. They are, of course, smaller. We also have the syringe with intraoral needle. So the needle is also having a protective cap. And obviously, we are going to look into different sizes of syringes and, again, those needles as well. The last, I would say, closure system that we can see on the slide on the right side is the syringe with luer lock tip. So here, the lock tip is going to allow us to insert the needle and then to turn a little bit the needle so it is going to be locked against the syringe. And obviously, we also, when we again offer this type of syringe, we also add a protective cap as well. Now, continuing with the container closure system, the physical and chemical characteristics of containers and closures can be critical to the sterility and stability of the finished product. So even though many of them may look similar, they can be actually made from different materials or have special surface treatments like silicone or stopper or ammonium sulfate or type 2 glass, for example. Now, as per the Chemistry and Manufacturing and Control Regulatory Affairs, one important task is evaluating how companies ensure their containers and closures meet specifications every time. Excuse me. So this includes checking that they consistently maintain quality and integrity throughout the product's shelf life. Now let's talk about the sterility testings. These are testings or alternatives like container closure integrity testing. And this testing should be done at least at the beginning and end of a product's shelf life to make sure this is still protected. It's also critical to confirm that the right tests are in place to verify the material's dimensions and overall quality of containers and closures. So if these aren't correct, the drug product could be compromised. So in short, getting this right is essential for ensuring product safety and effectiveness. Now here we are looking into the submodule 32P7 in the command technical document backbone, where we have to add the information related to our container closure system. So this is in module 3, submodule 2, P7, P standing for the product. Now, what is inside, in this document? So when submitting a drug application, it's important to provide a full description of container closure system. This means detailing each component, including the materials used, their specification, dimensions, and even drawings of the system. So the choice of materials matters. So there should be a clear justification for why each material was selected. It's also critical to consider any potential interaction between the product and its container or even the labeling, as this could impact the drug's stability and safety. So if a dosing device is part of the system, like an inhaler, it's essential to demonstrate that it consistently delivers an accurate and reproducible dose under conditions that mimic real-world use. So regulators want to see proof in this module that patients will receive the right amount of medication every time they use the product. So again, we're talking about the CTD module 3 to P7. Now here, for example, I'm giving you an example of what we add or write in the document that we're going to add into CTD module 3 to P7. So here is for product X, for example, we write the following. We write for our product, the primary packaging consists of blister strips. These strips are made of a laminated blister film, specifically what laminated to what, and are sealed with what. So importantly, the blister leading film meets the regulatory requirements outlined in 21 CFR 175.300 and of course 380, ensuring its safety for pharmaceutical use. So additionally, the film used in this container closure system complies with 21 CFR 177.1380 and meets USP class, and we write down which class, like 611 or 661, plastic standards. So moving to the secondary packaging, we write that the product X is housed in blister cards. So these cards are made of a material and are designed to be both tamper evident and child resistant. So this added layer of security helps protect the product while ensuring patients' safety. So this is a type of I would say information that we add in a regulatory document that we are going to prepare and then add into CTD module 3 to P7. Now let's talk about container closure related FDA warning letter. Why? That's because we need to be in full compliance with the FDA regulations. Now, it happens that after FDA inspections, some companies, since they are not in full compliance, are going to receive warning letters or letters 483, FDA letters 483, in order to understand, of course, helping them to correct the situation. And here, of course, they have to go to the CAPA, or corrective action and preventive actions. So let's go over a few FDA warning letters. So here we are talking about an FDA warning letter. So this is issued in May 31st, 2013, highlighting serious deficiencies in container closure integrity. Specifically, the firm failed to ensure that its container closure system provided adequate protection against external factors that could lead to product deterioration or contamination, and this is violating Title 21 of Code of Federal Regulation 211.94, paragraph B. Now, to make matters worse, between November 2018 and March 2013, 2011 to March 2013, the company received consumer complaints about at least 10 membrane leaks and 155 cases of improperly fitting blue caps. Of course, the information here is coming from the quality management system of the company, where quality assurance department has to receive all of the complaints from patients and consumers. They have also to do what we call a root cause analysis in order to prevent harm and, of course, to stay in compliance with the FDA regulations. Now, based on this warning letter, these critical defects directly impacted products' sterility and stability. So even more concerning in this letter is that this wasn't a first-time offense, it was a repeated violation, signaling a major compliance issue in that company. We can also have another case of FDA warning letter. In this warning letter, as of November 2nd, 2015, investigators identified critical current good manufacturing practice or CGMP violations that led to the classification of drug products as adulterated under Section 501, Paragraph A, Subparagraph 2, Subparagraph B of the FDCA. So one major issue was the failure to establish proper written procedures or SOPs ensuring batch uniformity and integrity. So these procedures should define in process control, testing and examinations for each batch as required by 21 Code of Federal Regulations 211.110, paragraph A. Additionally, the warning letter is stating that the firm did not ensure that container closure system provided adequate protection against external factors that could lead to product deterioration or contamination, violating Title 21 Code of Regulation 211.94 or Section 94, paragraph B. So these findings highlight significant compliance gaps that could compromise product quality and patient safety. Now, let's go over the container closure types and materials. So this image displays an ampoule containing epinephrine injection, USP 1 milligram per milliliter. You can see, of course, from the top, the conical tip. Down to that, you have the scored point, then you have the neck, and you have the body of the ampoule. Now, the primary packaging consists of a sealed glass ampoule. A critical component is ensuring product sterility and stability because it's a glass. So the glass ampoules are widely used for injectable medications due to their excellent barrier properties against moisture, oxygen, and light. Now, proper handling is essential, of course, to avoid contamination or breakage, as ampoules require careful opening techniques. Now, regulatory requirements for ampoule packaging ensure that the material meets USP standards and provides adequate protection throughout storage and use. Now, let's talk about vials. So this slide is illustrating various types of pharmaceutical vials and closure systems commonly used for injectable drugs. Now, as we see, glass vials show in different sizes and configurations, and they serve as primary packaging for liquid and lyophilized drug products. So these vials are typically made of type 1 borosilicate glasses, ensuring chemical resistance and maintaining drug stability and sterility. Now, you also see rubbers. So these are rubber stoppers that provide a secure seal while allowing for needle penetration for drug withdrawal. So flip-off seals and cream caps further enhance sterility by preventing contamination. Now, the selection of an appropriate container closure system is critical for maintaining product integrity throughout its shelf life. Now let's go over the blister packaging. So this slide is highlighting blister packaging, a widely used format for solid oral dosage forms such as tablets and capsules. Blister packs provide a protective barrier against moisture, oxygen, and light, helping to maintain product stability and shelf life. Now, the individual cavities in the packaging offer unit dose protection, reducing the risk of contamination and ensuring accurate dosing. So materials typically include PVC and PVDC or aluminum foil, selected based on the drug's sensitivity to environmental factors. Now, blister packaging is preferred for its tamper evidence feature, portability, and ease of use, making it a common choice for both prescription and over-the-counter medications. Now, continuing with the rubber closure, there are several rubber materials that can be used in pharmaceutical packaging. Now we can, for example, talk about butyl rubber and chlorobutyl rubbers that have the majority share of parenteral closure market. Now we also have siliconized rubber that have limited applications in pharmaceuticals as they are prone to tearing. Now, talking about pre-filled syringes, they are an increasingly popular drug delivery system. They are ready to use and come preloaded with medication, often with a needle already attached, as seen with insulin syringes. The pharmaceutical industry is continuously searching, finding new types of new materials that we can use in syringe. Pre-filled syringes. This is for being safer and being more efficient, and obviously more cost-effective delivery methods. Now, traditional multi-dose and single-dose vials, once the standard of vaccines and injectable, are gradually being replaced by pre-filled syringes due to their convenience and reduced risk of contamination. So we'll see this more and more, of course, on the market. Now, pre-filled syringes are widely used for delivering various drug categories, including vaccines, therapeutic proteins, blood stimulants, and erythropoietins. Additionally, there is a growing trend toward using plastic-based cyclopolyolefin, resin, and syringe manufacturing. So this material offers advantages such as improved strength, reduced risk of breakage, and enhanced compatibility with sensitive biologics. Now let's talk about parenteral products such as injectable medications, because they require specialized packaging to maintain their safety and efficacy. So these formulations are typically packaged in containers made of either glass or plastic. So the most common container system includes ampoules, vials, syringes, cartridges, bottles, and bags. And unlike other dosage forms, parenteral products are injected directly into body tissues. So they are bypassing the skin and mucous membranes. So because of this direct administration route, These products must be exceptionally pure. They must be free from physical, chemical, and biological contaminants to ensure patient safety. So this requirement places a significant responsibility on pharmaceutical companies to adhere to current good manufacturing practices during the production and packaging of parenteral dosage forms. So additionally, healthcare professionals must follow strict aseptic administration practices to prevent contamination and ensure safe dose. Okay, so now continuing with again the packaging of parenteral products, we're going to talk about the glass here. Glass is a key material used in the packaging of parenteral products due to its stability and resistance to chemical interactions. It's primarily composed of silicon dioxide with additional oxides such as sodium, potassium, calcium, magnesium, aluminum, boron, and iron, which influence its properties. So the United States Pharmacopoeia, or USP, classifies pharmaceutical glass into 4 types based on its chemical resistance and suitability for different formulations. So as we can see, type 1 is a neutral borosilicate glass. This is the highest quality glass, offering excellent resistance to leachables, permeation, and adsorption. We have type 2 is it is suitable for all types of parenteral formulations, including acidic, neutral, and alkaline solution. Now, let's go over type 2. For type 2, these are treated soda lime glasses. So this glass undergoes surface treatment to improve its chemical resistance, and it is suitable for buffered solutions, formulations with a pH below 7, or products that do not react with glass. We have the type 3, which is related to regular soda lime glass. This type has limited resistance to chemical interactions, and its primary use is for anhydrous liquids or dry powders. Now we have type number 4, which is general soda lime glass. This is not suitable for parenteral packaging due to its high potential for chemical interactions and leaching. And now let's go over the tests. To determine the glass type, the USP requires 2 tests. Test number 1 is powdered glass test that assesses the amount of extractable alkali from the glass. We also have water attack test. It measures the resistance of the glass surface to water. Now, proper selection of glass packaging is essential to ensure the safety, stability, and efficacy of parenteral drugs, and this is why we use, of course, the USP, the tests that, of course, are in the USP. Now, let's talk about the plastic containers. Plastics play a crucial role in parenteral packaging because they are lightweight, flexible, and resistant to breakage. Now, some of the most commonly used plastics are the following. One of them is polyethylene or PE, which is known for its flexibility and is often used in intravenous bags. We also have PP or polypropylene, which is heat resistant and commonly found in vials and syringes. We also have PVC, polyvinyl chloride, which is flexible and widely used for intravenous bags and tubings. We also have cyclic olefin polymers or COPs and COCs, which offer high purity and are used in pre-filled syringes and vials. Now, from a regulatory standpoint, USP number 661 ensures the safety and compatibility of these materials. Additionally, their barrier properties are critical in maintaining drug stability and preventing contamination. Now let's talk about syringes, because they are essential tools for injecting, withdrawing, or instilling fluids. They consist of a barrel, typically made of glass or plastic, with a plunger at one end and a small opening at the other end to hold the needle. Now, needle gauge refers to the outside diameter of the needle shaft. A larger gauge number means a smaller diameter. So we have common gauges that range from 13 to 27. So again, I repeat, a larger gauge number means a smaller diameter. We also have subcutaneous injections that usually require 22 or 25 gauge needles. For example, now intramuscular injections use needles between 19 and 22 gauge. For compounding parenteral products, 18 to 20 gauge needles are commonly used. Now let's talk about the container closure attributes. Now here in this guidance, we are able to see again the type and extent of information to provide depending on dosage form and route of administration. Excuse me. So here we can see on the first column the degree of concern associated with the route of administration. So we have the highest, high, and low. And then we have the likelihood of packaging component dosage form interactions. So for example, if we have a high likelihood of packaging component dosage form interaction, the definition is, I would say, sorry, the forms or the closures are, for example, drugs for inhalation aerosols and solutions, injections and injectables, and suspensions. So we see very well that there is a likelihood of packaging component to be in interaction with those products. But where we may have less interactions would be for oral tablets and oral hard and soft gelatin capsules. So we understand here that the extent of information will depend on the likelihood of packaging component interacting or being in contact with the container. So now let's go over the container closure, the suitability for the intended use. So every packaging system should be suitable for its intended use. It should protect the dosage form, it should be compatible with the dosage form, and it must be composed of materials considered safe for use with the dosage form and the route of administration. So a description of the tests that we are going to do, obviously, is also needed when we are going to submit our application to register our drug. Now, related to the container closure protection, a container closure system should provide the drug protection from factors like temperature, the light, of course, we have also pressure, etc., that can cause degradation in the quality of what dosage form over shelf life. Humidity is also another of these factors. So common causes of degradation are exposure to light, loss of solvent, exposure to reactive gases, absorption of water vapor, and microbial contamination. So not every drug product is susceptible to degradation by these factors. So not all drugs products are light sensitive. Not all tablets are subject to loss of quality due to absorption of moisture. And obviously sensitivity to oxygen is most found with liquid-based dosage forms. So this is why we need to conduct studies. So here we talk about laboratory studies that can be used to determine which of these factors actually affect, again, the quality and the safety of our product. Now, in the guidance for industry for the container closure system for packaging human drugs and biologics, we can also see this table. This is a table related to the protection. So on the left side, we can see the route of administration or dosage and dosage form. And on the right side of the table, we can see the type of protections. And obviously, underneath, on the right side of the slide, we can see each of the abbreviations is standing for what type of protection. For example, for inhalation aerosols and solutions and nasal sprays, we have to look into the following protections, L, S, M, W, and G. It means that for testings, we need to provide results of testings, suitability testings that are related to 1, protection from light if appropriate, protection from solvent loss or leakage, protecting sterile products or those with microbial limits from microbial contamination, protecting from water vapor if appropriate, and protecting from reactive gases if appropriate. So now we understand that now, for example, if you're looking to a last item on the left table, our question would be, okay, what are the suitability testing that we need to conduct in order to ensure the protection of our products related to oral tablets and oral hard and soft gelatin capsules. So as we see based on this guidance for industry, we will see that for the oral tablets and oral gelatin capsules, we need to undergo or perform suitability testing for, again, the items L and W. It means we have to make sure that our container closure system is protecting the product from the light, and W is protecting from water vapor. So now we understand that this is a guidance for industry. If now you look into the table on the left side, if your pharmaceutical product is one of the, the products, or it's using a route of administration, as we can see on the left side of the table, then you have to look into the protection that is required and then to come up with the results of this suitability testing that have been performed, and then you add them into module 3 to P7, sorry, that we already looked at in the CTD format in the module. So these are the tests that you need to have in order to be able to be in compliance with the FDA regulations for the protection of your product. Now, talking about container closure and compatibility. So packaging components that are compatible with the dosage form will not interact sufficiently to cause unacceptable changes in the quality of either the dosage form or the packaging components. So we can talk about the examples of the interactions, like loss of property due to absorption or adsorption of the drug substance. We can talk about reduction in the concentration of an excipient due to absorption or adsorption or leachable induced degradation or precipitation. In addition, it may be changes in drug product pH, discoloration of either the dosage form or the packaging component, and finally, increase in brittleness of the packaging component. Now, coming back to the guidance for industry, we are continuing with the compatibility and the suitability testing related to compatibility. So the same philosophy here applies, the same requirements. It means that if now your drug is using any of the route of administration, or it has any dosage form as indicated on the table on the left side, then you have to undergo suitability testing related to compatibility. So here we have 3 types of suitability testing: case 1C, 2C, and 3C. Now I'm going to refer to the table on the left side, and I'm going to look into the first row only. If your product is again using inhalation aerosols and solutions, or this is one of the nasal sprays. In this case, you need to undergo a suitability testing related to the compatibility. In this case, you have to go to test 1C. It means liquid-based dosage form that conceivably could interact with its container closure system component. And again, this applies to other routes of administration or dosage form that you can see on the left side. So now here we understand that we need to prepare and to have adequate documents or results of the tests as per the guidance for industry that we are seeing here. Now let's talk about the safety. So container closure safety. So packaging components should be constructed of materials that will not leach harmful or undesirable amounts of substances to which a patient will be exposed. Now, safety is especially important for packaging components which may be in direct contact with the dosage form, but it is also applicable to any component from which substances may migrate into the dosage form. Now, we understand now that we need to have testing approaches that will depend on the route of administration and likelihood of interactions between the component and the dosage form. So here, in the same guidance for industry document, we have again very similar tables, but this time this is related to the safety. And on the table on the left, we can see again different routes of administration as well as the dosage forms. And we have the compatibility testing or that we need to again perform and then to provide the results of those testings to FDA when we are going to apply to register our drug. Now again, I'm going to choose the same example, which is on the table on the left, on the first row, which is inhalation aerosols and solutions and nasal sprays. So the compatibility here is 1S. So this is for the testing. Now, what is 1S? This is case 1S that you can see on the right side of this slide. So it's typically provided are USP biological reactivity test data, extraction, toxicological evaluation, limits on extractables, and batch-to-batch monitoring or extractables. So now we understand that if we are dealing with, again, products related to the first row, we will need to conduct suitability testing, and we will need to provide the results of those testings as per case 1S that we can see in this guidance for industry document. And of course, we have case 2S, case 3S, 4S, 5S. Again, each of those suitability testing will be different, again, depending on the route of administration or the dosage form of your product. And again, you will need to refer to this guidance for industry. Now, talking about the performance, a container closure system does more than to contain the dosage form. When evaluating performance, there are two major considerations that are functionality and drug delivery. Now, for the functionality, the container closure system may be designed to improve patient compliance. For example, a cap that contains a counter, minimize waste, for example, a two-chamber vial or an intravenous bag, improve ease of use, for example, a pre-filled syringe, or have other functions. But now for the drug delivery, it refers to the ability of the packaging system to deliver the dosage form. Now, when we again refer to the guidance for industry, on the left side again, we will see the routes of administration, the dosage forms. So again, I'm going to refer to first row, inhalation aerosols and solutions and nasal sprays. So we see that the results that we need for suitability testing are here, again, the case 1D, which means frequently a consideration. So we need to do the performance test if we are having products, again, in the class of inhalation aerosols and solutions and nasal, sprays. So we look into the functionality and we look into the delivery. This is frequently asked. Now, for example, for case 3D, oral tablets and oral hard and soft gelatin capsules, we are not asking, it's rare, or we may not ask for suitability testing for the delivery and for the functionalities because of the route of administration or the dosage form. Now here we can see overall what we have in the guidance for industry as of May 1999, container closure system for packaging human drugs and biologics. So on the left side of the table, we have the route of administration and the dosage form, and then we are looking into testing for the suitability, where we have the next 4 columns, one related to the protection, then to the compatibility, to the safety, and then to the performance and drug delivery. So this is a table that you will see in the guidance for industry, and the previous slides that I explained to you, they were just to prepare you to understand what are the requirements for the suitability testing and what are the documents that we need you, of course, to prepare and to add into the module 3 of the common technical document when you are going to apply to register your drug. Now, again, it may be to FDA, you may send it your new drug submission to Health Canada, or your MMA or marketing application authorization application to Europe. In any case, module 3 of the CTD is universal. So wherever in any country where they are again following the ICH guidelines, if you would like to register your product, you will need also to comply with this regulation, and the regulation is allowing you to know about what are the tests that we are asking you to perform in order to be in compliance with the container closure system for packaging human drugs and biologics guidance for industry. Now here, this is again what you can see when you open the document. So here you have again the information that should be submitted in an original application for any drug product. So as you see, the first type of documents is related to the description of your product. The second important section or type of documents or the information is related to the suitability. So here we talk about the protection, for example, from light or from reactive gases, etc. We also ask you to give information related to the safety of your product, so chemical composition of all plastic, elastomers or adhesive, etc. Looking into the extraction or toxicological evaluation studies as appropriate, so if applicable, looking into appropriate USP testings that you performed. So all of this or this package of documents are all going to be related to the suitability tests related to your product, and again related to the closure systems. You also need to provide information on the quality control. So for each packaging component received by the applicant, you are going to provide FDA or Health Canada or EMA with the applicant's tests and acceptance criteria. And again, here you are going to refer to USP, and you are going to refer to the suitability tests that are already posted or added or stated into this guidance document. Now here when we talk about the description of the guidance documents, we are talking about the general description of the entire container closure system that you need to provide. This is again the first item that we looked at in the previous slide. The next item that we saw was about the safety. So the safety is again going to be added into again a second, I would say, section of your report, where you give information about the safety and the quality. So for each packaging component received by the applicant, applicant's tests and acceptance criteria must be provided. Then dimensional or drawings and performance criteria must also be provided, and method to monitor consistency in the composition of the container closure must also be provided. Now for each packaging component provided by the supplier, you also need to provide the manufacturer's acceptance criteria for release. This is as per CGMP or GMP. And then a brief description of the manufacturing process. So now we understand that when we are going to prepare the information for the container closure section in the CTD module 3, we will need to follow the requirements of being compliance with this guidance for industry, then to look into what are the suitability tests that are required, and then to look into what are the types of information that we have to prepare and to add into that sub-module in order to obviously giving complete information to a regulatory authority. Now let's talk about the container closure testing methods. These are tests that are performed for the suitability testings. So we need to use those tests, then to document the results, and then to add them into the, again, the information that we are going to submit in our module 3 related to container closure testings. So talking about the closures for pharmaceutical preparation, before using a closure in a vial or bottle, the closure must be assessed to determine if it's suitable for use with the product that will be filled into the container. So the pharmaceutical manufacturer should consider the following questions. So is the product absorbed by the rubber? Does the rubber react with the product and leach out impurities? At which temperature range is both closure and product stable? How effectively is the seal integrity? What happens when the product and stopper are stored together over time? Now, when we refer to USP 1207, it's stipulating that container closure integrity testing can be performed in many different ways. All methods have advantages and disadvantages. Now, some containers, such as ampoules, require 100% integrity testing. Now, recent changes in USP 1207, which became official in 2016, describe several methods that user can experiment with. So the proposed updated removes the requirements to compare the results of other container closure integrity tests to the microbial ingress challenges. Now, when we look into the USP 1207, we have two major categories of tests. We have one category of tests that we call deterministic, and we have another category of tests that we call probabilistic. So what is the deterministic tests? These are tests which are less subject to errors and provides quantitative results. But how about probabilistic tests? They involve more uncertainty in assay results and include more traditional methods. And that's because we may not be able to necessarily, again, test two different products with exactly the same method. That's why we have the deterministic and probabilistic methods. Now here, we are able now to look into the table where on the left you have the deterministic methods, and on the right side you have the probabilistic methods. As you see, the deterministic methods, they are reproducible. They are sensitive because we use equipments as well, or sensors, for example. They are also highly instrumental because we need to have sensors and other equipments. They are also allowing us to have qualitative test results because we can measure something accurately. And of course, it needs minimal test sample preparation or manipulation, and it has a lower risk of error. Now, we can look into probabilistic methods. They may not be reproducible. They are usually insensitive. We usually don't use we use little or no instrumentation again for the tests. They are usually qualitative, and we just do the interpretation of the results. But again, they are insensitive, and they are considerable test sample preparation and or manipulation, and they have a higher risk of error. Now let's go over a few selected tests. Here, let's talk about the probabilistic test, which is the bubble point test. So what we do here is that the test package under test is pressurized and immersed in a bath containing water or water and a surfactant like polysorbate 80. Now a differential pressure is applied on the container. The container is observed for bubbles, so positive leak test results in air bubbles. Again, as you see, we are not using an instrumentation. It's not a sensitive test, and again, it may have a high risk, but this is a probabilistic test that we can, again, perform for this type of product. We can also talk about another probabilistic test, which is about the tracer gas leak method, or we call that a Stephen mode. So in this test samples are flooded with the tracer gas like helium, then checked for leakage by scanning the outer package surfaces using a vacuum wand. An advantage is that the sensitivity of the method can be increased by increasing the tracer gas pressure, but the disadvantage is that the leak rate cannot be measured exactly, and sensitivity is limited by helium content in the air because it's about 5 parts per million, so which produces, of course, a background. However, we still use some tests, of course, some detectors, but as you see, this is a probabilistic test, an acceptable test, obviously, but this is a probabilistic test. Now, we can also now talk about deterministic methods. One of them is electrical conductivity and capacitance test. Another one is laser-based gas headspace analysis. We have mass extraction test, pressure decay test, tracer gas or vacuum mode test, and vacuum decay. In the next slides, I'm going to just go over a few selected of these deterministic methods. Now let's go over the first deterministic method, the selected one, which is the high voltage leak detection. Now it consists of a set of electrode probes which scans a sealed container made of non-conductive materials. So materials can be glass, plastic, or polylaminates. So if a pinhole, a crack, or seal imperfection or other defects is present, there is a differential on the current flow or the voltage indicating a breach of the container closure. So applications include pre-filled syringes, liquid filled vials, and ampoules. The benefits include high level of repeatability and accuracy, efficiency, non-destructive, non-invasive, and no sample preparation. Another example of deterministic test is a laser-based gas headspace analysis, as you can see at the bottom of this slide. Now, typically performed using non-contact methods, then this method is non-destructive and provides quantitative results. So near-infrared diode laser light passes through the gas headspace region. Light and is absorbed as a function of gas concentration and pressure, and absorption information is processed using phase-sensitive detection techniques. Now, the procedure can be used for lyophilized products or oxygen-sensitive liquid products. It's rapid and allows 100% inspection of oxygen-sensitive products or products packaged under vacuum. Now, the key parameters include headspace volume, package temperature, headspace pressure, and vacuum and sensitivity. Now, this table is again showing, again in just one slide, the container closure integrity tests. So On the left side, you have all of the tests such as helium mass spectrometry, high voltage detection, acoustic imaging, bubble test, residual gas ionization test, liquid tracer test, and of course you have several other tests as well. Now in the second column from left to right, we have the principles where for helium mass spectrometry, we are placing helium inside or outside of the container, and migration of helium is detected by mass spectrometry. For the high voltage detection, we already explained that in the previous slide. We can have acoustic imaging where we use ultrasonic energy that is focused on sample submerged in the water. So we look into the echo patterns that will produce an image. We also have the bubble test that we already explained. We also have residual gas ionization test where we use a high voltage field which is applied to vials sealed under vacuum, so the field causes residual gas to glow. And we also have liquid tracer test where the package is immersed in solution of tracer chemical or dye, recolor, pressure or vacuum or temperature cycling to improve sensitivity, and we see leakage detected visually or mechanically or with a transmitter, for example. So with a receptor, sorry. So we have advantages and disadvantages, but again, the aim of this lecture was just going over the container closure systems and obviously going over the tests that are required and the regulations applied in order to show to regulatory authorities that we have been doing the test as per the regulations in place. Now, in summary, container closure integrity testing can be performed in many different ways. So each method has advantages and disadvantages. We looked into USB 1206, where it was dividing container closure integrity tests into two major categories. We looked into deterministic methods or tests, and we also looked into probabilistic methods and tests. So now a trend in container closure integrity testing is a shift from probabilistic methodology to deterministic methodology. This is why you're going to see several types of methods as well for testing the safety and the functionality, of course, and the safety and the delivery modes, of course, of the closure systems. Now, the ultimate proof of suitability of the container closure system and the packaging process is established by full shelf life stability studies as well. So I would like just to thank you so much again for joining me for today's class. I hope that you enjoyed the content of a very interesting topic for today. I wish you all again a great day. Thank you so much.